Hi, I'm Federica, and today I would like to share with you the great experience that is being part of the synBIOcarb network. synBIOcarb is under the European program Horizon 2020, and it is made of 15 PhD students based in different European countries. All of us contribute to add a brick of knowledge in the field of synthetic glycobiology. I am based in France, more specifically in the city of Orléans. This is a very famous place on the Loire River, mostly known to have been the theatre of the battle where Jeanne d'Arc defeated the English army. She is the symbol of the city, and she is basically everywhere. But let me start from the beginning. A few years ago, I graduated in biotechnology at the University of Milano Bicocca. After my master, my goal was to do research that could be easily applied. So in synBIOcarb I have the chance to do it because I am carrying out my project in the innovative company Glycobiology. Glycobiology was founded in 2005 and it's been the first company to start Lactin Array research, application and services in France. I am sure you are now thinking, yes, this seems very cool, but what is a Lactin Array? For starters, lectins are carbohydrate binding proteins. They were discovered more than 100 years ago in plants, but they are known to be present in all living organisms that produce glycans. Don't get confused, they are not antibodies. They recognize sugars with their carbohydrate recognition domain, the CRD. They are not enzymes and they don't catalyze a modification in the glycans. Moreover, lectins can recognize different linkages between two carbohydrates within a glycan structure. So, for example, let's say we have a lectin array where lectins are immobilized at the bottom of a 96-well plate. Then we have two possibilities. We have the direct binding model where the sample is applied and it needs to be previously labeled by biotinylation or fluoresceinylation to allow the readout with a spectrophotometer. The second one is the inhibition model, where the sample doesn't need to be labeled. It is applied to the plate, and at the same time, a fluorescent or biotinylated glycoprotein or neoglycoprotein, known to have high affinity for the lectin, is applied too. The intensity of fluorescence or absorbance is again measured thanks to a spectrophotometer. So what we see is that there is a competition for the binding to the lectin between the sample and the labeled molecule. The intensity of fluorescence, or a colorimetric reaction, will tell us the percentage with whom the labeled molecule inhibits the binding of the sample to the lectin. It is a sensitive, high-throughput screening of carbohydrates and glycoconjugates, and it is very handy because it does not require prior chemical or enzymatic digestion of the sample or a huge expertise in managing the technique, like for example what happens with mass spectrometry. Moreover, in the inhibition model, there is no need of pre-labeling of the sample. My main goal is to purify lectins coming from natural sources, or recombinant lectins expressed in genetically engineered strains of E. coli. The target lectins recognize non-human glycosylation, like alpha-gal and glycolyl-neuraminic acid epitopes. They can be found in terminal positions of glycans on some biological medicinal products prepared in non-human genetic engineered systems. Humans have circulating antibodies against these glycans, and potential antigen-antibody response could be deleterious and could affect therapeutic efficiency. Building a lectin array specific for this glycosylation will allow a quality control screening during the early phases of production of recombinant biotherapeutics products. In synBIOcarb, collaboration among the 15 of us is crucial. I spent last January at CERMAV in Grenoble, where I learned more on expression and purification of recombinant lectins. So, if you are interested in knowing more about synBIOcarb and my work, I'll leave some link in the description below. And don't forget to follow us on Twitter for the latest news.